Today, I'm going to talk to you about the really important concept of something called glycolysis. And so glycolysis is an anaerobic process used to produce energy from carbohydrates or from glycogen. Now, there's many videos that you can watch about this, so I would absolutely encourage you to watch more than just mine. But also make sure that you understand why we're doing this. We're doing this in order to get energy. Okay, so a 6-carbon glucose, we're going to break it down first into 2 pyruvate molecules. Okay, pyruvate are 3 carbon molecules that we can do other cool stuff with that we're going to look at later. But this is the beginning of our processes, okay? Now, guys, this is a 10-step transformation. It is not for the lighthearted, but you do it all the time inside of your body, so you could totally do this, right? In the first 5 steps, are going to actually use energy. Just because, you have to use energy to get stuff, and we're going to use ATP and turn it into ADP. But the next 5 steps produce ATP, and they produce this cool thing called NADH. Now, NADH, we talked about last time, there's cool stuff that NADH will do at the end, and we end up getting energy out of that too, or we can do other things with it that are super cool. So there's a lot of options here. As an overall process, what we find is that we can use 2 NAD plus glucose and 2 ATP to produce 4 ATP, 2 NADH, 2 ADP, and 2 phosphate. So that's what this means, this PI down here. So your net gain in glycolysis is to get 2 ATP and 2 NADH out of it. Okay, that's a lot of stuff. Let's look at where we get stuff from, how we do it, and what glycolysis is. Let's look at those 10 steps. So we start again by using glucose. So as you remember, this is alpha D glucose, and that's what we're starting with. So if we start with glucose, reaction number 1 uses enzymes. Now I put the enzymes here to the right-hand side of these reactions. And notice that I put this little thing where you use this and it produces that to the side. So what that's saying is you use one of these things, and in the end, it also produces the thing that's coming out of that reaction there. So we're going to use an enzyme called hexokinase, and we're also going to produce ADP and what we see down here. So glucose 6-phosphate. So what we just did in this first reaction here is that we transferred the phosphate from ATP over here to this guy, okay? So that's why he's now glucose 6-phosphate, which totally makes sense. At least I hope so. And so next, what we're going to do is we're going to use the next enzyme, which is called phosphoglucose isomerase, and it's going to isomerize glucose 6-phosphate to fructose 6-phosphate. Now remember, isomerases and isomers mean that we just moved molecules a little bit around. So what ended up happening is this carbon right here just kind of got kicked up out of the ring and is now facing upwards. Let me move this on down here so we can discuss this a little bit more. Okay, in reaction 3, we again are going to use ATP producing ADP. So we're going to use another bit of energy and another phosphate, and we're going to use phosphofructokinase, which is going to basically phosphorylate the second or the first position here on fructose. So we end up with fructose 1,6-bisphosphate. See how this guy comes in right there? So now we've got phosphates on both sides of this fructose. So reaction 4 is actually really kind of cool. What ends up happening is this aldolase comes in and it cleaves both of the glucose into 2 and 3 carbon phosphate isomers. Okay, now what ends up happening is we end up with this guy, which is known as glyceraldehyde 3-phosphate, and we also end up with this guy. Well, here's what's neat. In the fifth reaction, the one that's over here uses another enzyme called triose phosphate isomerase to make this, which is the same thing as what this guy is. So these two things are the same. And so you end up with two molecules of glyceraldehyde 3-phosphate when you go into reaction 6. So when you start to the next 5 reactions, you almost have to consider, hey, I've got 2 of these for every glucose that I started with. So you'll start to see the numbers sort of double, and that's the reason why. So entering this steps 6 through 10, these are where energy is actually produced. So keep in mind we have 2 molecules of glyceraldehyde that we're playing with from the previous screen. So this is what we start with, again, is glyceraldehyde 3-phosphate. So this is where we start. And you'll notice if you want to go back, this is the same thing that we just finished with, but we ended up with 2 of them, okay? So here's where we start to use NAD and produce NADH in this reaction, okay? This is going to use an enzyme called glyceraldehyde 3-phosphate dehydrogenase. And what this guy does is he adds another phosphate right up here, okay? So now we have phosphates and 3 carbons, and we've got a COH there in the middle. And so what then happens is ADP comes around and transfers one of those phosphates to ATP using phosphoglycerate kinase, which is another enzyme. So in that process, what we're doing is we're going to convert 1,3-bisphosphoglycerate to just 3-phosphoglycerate. So see how we got rid of the phosphate that was up here? I just want to point out the fact that this is different than what we started with originally. We have a COO here versus in the beginning, that's not what we started with. So these are going to be 2 different molecules, and therefore they will react in different ways, okay? I'm going to go on down the page here and just go right there for right now. Okay. So that was a transfer of phosphate to ATP in reaction 7. And in the next reaction, we're going to use phosphoglycerate kinase in order to isomerize that product to get 2-phosphoglycerate. So that's what we have right here, okay? So what's changed in these 2? Well, we actually moved the location of where that phosphate was. See how down here, that's on the last carbon? Right here, that's on the second carbon. So we changed where things were located again using that isomerization reaction. Okay, so now we have a phosphate on that second carbon. We can either add or remove water to go through a dehydration using enolase as our enzyme here, and this produces a CC double bond right down here, okay? So I'm just going to point out where the water is coming from. We're going to get this guy out along with the hydrogen that's right here, and I'm sorry, I did not mean to write that there's a hydrogen right there because there's not enough bonds to have that hydrogen right there. Okay. So in this, we now have a product where we can use ADP and remove another phosphate group with pyruvate kinase in the final step, which is a transfer of phosphate to ADP. Now, in the end here, what we end up with is pyruvate. And this is incredibly important when we move on to the next step, okay? And so we're going to use pyruvate to enter the next part of metabolism in just a little bit. Now, let's talk about this. Reaction 6 to 10, what we end up producing is 2 ATP and 1 NADH, for each of the glyceraldehyde from step 5. And remember, there's 2 glyceraldehyde that we use for step 5. So that means that our net for 1 molecule of glucose is 4 ATP and 2 NADH. But if you remember, steps 1 through 5 use 2 ATP. And so what that means is what we get out of glycolysis for 1 molecule of glucose is 2 ATP, 2 NADH, and 2 pyruvate, just like I told you at the very beginning of this video. Now, guys, I have no anticipation that you actually remembered every single bit of what I just said there. You're going to have to go back and review this a few times. You're probably going to have to go back and review several other videos too. That is okay. That is normal. Be able to identify different steps, what's happening, describe what's happening, be able to tell what types of enzymes you have here and how they're operating and what they're doing. Those are important things to know. Know how much energy is produced, generally where that's produced, and you're probably okay. So before we move on to the next area, let me go ahead and point out, fructose and galactose are also put through glycolysis, even though it's not glucose. They're first converted to sugars that can enter this glycolytic pathway at different points in time. So that's one of the cool things about having so many steps in glycolysis is that you can actually move in and have other things come in and maybe step 2 or step whatever it may be. The other really awesome thing about glycolysis is that it can be regulated, and the way it's regulated is super awesome. At least I think so. So your body needs ATP more at some times, less at others. So specifically steps 1, 3, and 10 are not reversible reactions. That means that they can't go forward and back and forward and back. So you can control things a little bit more based on how that enzyme is behaving. So let's go through those. In step 1, we have hexokinase. This guy is inhibited by a buildup of its product. So allosteric control, you know how we talked about that before? That's what's going on here. So if you produce less glucose in glycolysis, then the production is going to slow down. Okay, if that is going to be buildup of whatever glucose that is. Think of this as a factory. If you have like a candy factory and you're producing lots and lots of candy and you run out of wrappers, you need to slow the machine down so you can put more wrapper stuff into the machine and it can keep going that way and actually fill things. And so like when the machine is going crazy and you have such extra candy that's going through, then what happens is it like the machine says, hold up, wait a second, let me fill this up. And then you can keep on going with it once you don't have an excess of that candy anymore. In reaction 3, we use phosphofructokinase. So that's going to be reaction 3. It's another allosteric enzyme, a major control point of glycolysis. Too much ATP is going to inhibit that enzyme and say, wait, stop for a minute. If you have lots of ADP or AMP, that will actually activate that enzyme. So that's another cool part of it. And finally, reaction number 10 uses pyruvate kinase. It's also an allosteric control and inhibited by high levels of either ATP or coenzyme A. Now, we talked about coenzyme A last time, but we didn't really talk about all the stuff that it does. So we'll come back to coenzyme A a little bit later. Again, this is probably a lot. It is absolutely okay to review this more than once, and I would actually very much encourage that. Take your time, write this out on your own, and try to see what types of steps you can see in there. Thank you very much for joining me, and I'll see you soon.